My name is Alexandra Cook. Today I'm going to be discussing the topic gene therapy. And gene therapy is a technique that, in practice, can be used to cure severe and even deadly genetic disorders. About 1 in 10 Americans and around 30 million globally have a genetic disorder. That many people are suffering with genetic disorders. And 95% of these disorders have no effective treatments whatsoever. We can only basically help them survive a little more comfortably, and a lot of times their lives are faced with a lot of difficulties and are often shortened because of these disorders. Many of these genetic disorders are hereditary, so they can be passed from parent to child and through the family lines. This is done by, even if you don't express the gene, you could have a recessive gene, which paired with a partner's recessive gene could risk seeing it expressed in a child or an offspring. So, we are going to discuss today how genes work and how mutations cause these genetic disorders. Then I'm going to talk about how gene therapy will work and what they seek to accomplish, but I'm also going to discuss the problems that we see in gene therapy and why it's not yet quite successful and why we don't use it yet. So the first question we must answer is, what is a gene? What does a gene do for the cell, and how can it cause these genetic disorders? Basically, the gene is DNA. It is a sequence of DNAs in a nucleotide sequence. As you can see, there are 4 different types of letters. They just repeat, go down the line. It's basically the blueprint which instructs the cell which building blocks to use in order to build proteins, which help us function, determine our individuality, things like that. There are 4 different nucleotides. Each letter is going to be paired up with a corresponding specific letter. And this is how the gene code is read by the cell. As we see here, here's our DNA sequence, and there will be 3 corresponding letters. This is called a codon. It's read by groups of 3. And attached to that group of 3 is going to be an amino acid. Basically, it just keeps going down the chain. As you can see, here's 3, an amino acid, 3, amino acid. It reads the entire DNA to create this long chain of amino acids, which will make the proteins. So as you can see, the DNA is used as a blueprint to tell which ones will go where. I gave a little example here. For example, this sequence here is CATDOG, CATDOG. Now, if we were a cell, we would be reading this by 3s, right? So, we're going to read it by cat, dog, each group of 3. Now, each of these 3-letter words has a meaning to us. We can correlate an understanding with this. Now, the problem is what happens if we add or delete a letter? We're going to show an example of adding a letter to this little sequence here. We're going to add a G right there in the middle of it. Now, remember, we're going to be reading these in groups of 3. So we got CAT, DGO, GCA. These words here make absolutely no sense. So this one letter threw off the entire sequence. And this is basically what a genetic disorder is. Mutations will cause either adding of a nucleotide or letter or deleting one. And so by doing this, it's going to throw off the entire sequence. And wrong amino acids are going to hook onto these because this doesn't mean anything. So it's going to code for a wrong amino acid, which in turn will make dysfunctional proteins that are very faulty and bad for you. What gene therapy does to fix and cure these genetic disorders is they're going to take a sample of the DNA, they're going to code it, and they're going to find that problematic little nucleotide. Take it out, okay? It doesn't belong there. We have our little sequence here fixed. Now the blueprint's correct, and we'll create a right protein, right? In order to do this, we need to insert our sample of the correct DNA into a vector, which the best use is a virus, because a virus will attach to a cell and directly insert its DNA, in this case the good DNA, into the cell with the bad DNA, and the good DNA will hopefully just replicate and, cause healing. But the problems that we see with gene therapy are a couple of things that we run into, because our cells are very, very picky. They don't just let anybody in. So, we see that cells are assigned specific coding jobs. Each cell needs a certain type of blueprint, like there are certain cells for hair color or eye color or blood type. If we insert a blueprint gene for hair coloring into the blood type 1, it's not going to work. So we need to figure out a way to get the virus to attach to a certain type of cell that are targeted. And another problem is that a virus, the outside part of the virus, when it connects to the cell, can trigger a response from the cell. It can be like, oh no, somebody's invading. So in order to get rid of the little virus, it will cause an immune response, which will cause the person to have fever, whatever type of complications, and could even cause death. So this is definitely a big issue. We have to figure out a way not to alarm the cells when inserting this DNA. Another problem is that the bad DNA might copy faster than the good DNA. Now, the cells have already been wired with the bad DNA, and it is possible that even after giving the good DNA, that the bad DNA still might just replicate faster and faster, and the good one may just die off and not be effective before we can see any good happen in the patient. So as I've discussed here, some of the problems with gene therapy and but how we could see it work possibly one day. So faulty genes, obviously they're what cause the genetic disorders, and it's very important for the public to know, because the scientific community needs the support and research, as we have many things to figure out. We need bright minds to enter into the field and do research, and we also need the public to be informed, and we need their support. Because genetic disorders can affect you. They can affect the people that you love. And we are so close to finding a cure, but we need you to spread the word, and we need your support. Thank you for listening.